breath-hold divers can stay underwater for long periods even when oxygen levels become very low. We know that diving mammals such as seals have special muscle adaptations that help them conserve oxygen. However, it is still unclear whether human breath-hold divers develop similar adaptations, so this knowledge gap is what this study aimed to address. The objective of this study was to determine whether elite breath-hold divers have muscle adaptation that help conserve oxygen. To investigate this, the researchers compared the elite breath-hold divers with non-diving athletes. They examined mitochondrial function and oxygen use in skeletal muscle to identify adaptation that may explain the divers' exceptional performance.
I am now focusing on the technology framework. Let's start with how this muscle adaptation were actually measured in the lab. To study what happened inside the muscle tissue of elite breath-hold divers, 5 key technologies were used. First, oxygraph 2K high-resolution respirometry was used to measure exactly how fast the muscle cell breathes and consume oxygen. Second, flow LED 2 modules were attached to track cellular stress by monitoring mitochondrial hydrogen peroxide levels. Third, Western blotting was performed to identify, isolate, and count the specific protein essential for the muscle oxygenation. Fourth, spectrophotometry allows us to measure metabolic enzyme speeds to see how quickly energy is processed. And finally, gel electrophoresis map out the muscle tissue to identify the exact muscle fiber types present in the divers.
The most interesting finding is actually a surprise. We expected breath-hold divers to show higher mitochondrial capacity in their muscles, similar to diving mammals like seals. Instead, we found the opposite. So based on the paper, divers had lower mitochondrial oxygen consumption during both leak and maximal respiration compared to controls. And what makes this significant is that this happened even though enzyme activity and mitochondrial protein content were the same between the two groups. And that tells us that adaptation is not about having fewer or different mitochondria. It's about the mitochondria functioning differently at an intrinsic level. And this is the first time this kind of adaptation has been shown in humans. Before this, it was only documented in diving mammals. And it also revealed an interesting trade-off. Divers tended to produce more reactive oxygen species during this low oxygen state, linking oxygen conservation to oxidative stress. So yes, this does add new knowledge. It opens up a whole new angle on how the human body might adapt to hypoxia at the muscle level.
This research shows that elite breath-hold divers develop special mitochondrial adaptations in their skeletal muscles. These adaptations allow their muscles to consume less oxygen, especially during leak respiration and electron transfer system or ETS capacity. The emerging trend here is that researchers are no longer focusing only on aerobic performance. Instead, there is now more focus on mitochondrial efficiency and hypoxia tolerance. In simple words, performance is not only about having more oxygen, but also about using oxygen more efficiently. This is important because breath-hold divers need to survive in low oxygen conditions during prolonged apnea. Their muscles appear to reduce oxygen usage, which helps conserve oxygen for the vital organs such as the brain and heart. This adaptation is also similar to what can be seen in diving mammals such as seals. In the future, these findings may help research related to oxidative stress, isomia production, and hypoxia-related disease. However, the exact molecular mechanisms are still not fully understood. Therefore, more studies are needed to explain how these oxygen-conserving adaptations actually happen.
One key limitation is the very small sample size. Only 8 elite breath-hold divers and 8 controls. Elite free divers are a rare population, so it's hard to recruit large numbers. But this also means individual variation, like training background or genetics, can easily influence the results. Because of this, the findings may not fully generalize to recreational free divers or the broader population. and larger studies will be needed to confirm these adaptations are consistent.
Moving on to the conclusion, the ultimate takeaway from this research is that elite breast hole divers experience an incredible and protective biological adaptation. We found that divers possess a unique muscle changes that actively force their body to lower its oxygen usage. When underwater, their muscle cells switch into an ultra-efficient metabolic mode, essentially putting the skeletal muscle into a low-power mode. By shutting down high oxygen consumption in peripheral muscle, the body actively saves its limited oxygen supply. It then strategically redirects that saved oxygen straight to the most critical organ for survival, the brain and the heart. This proves that with training, human breast hole divers can successfully adapt to underwater environment using the exact same oxygen saving mechanism seen in marine mammals, such as seals.